The Institute of Forest Genetics and Tree Breeding under the Indian Council of Forestry Research and Education is a premier institute in the country which conducts research on genetic improvement and conservation of forestry species. The institute has more than 2 decades of experience in conducting DNA research. Deoxyribonucleic acid, which is popularly known as DNA, is a genetic material in most living organisms except few viruses. It can store up to 25 gigabyte of genetic information in just 1 inch. In forestry sector, DNA research has several applications which includes timber forensics, molecular breeding, genetic diversity assessment, species and hybrid validations, genetic engineering, and so on. In early 1990s, isolation of genomic DNA from plant tissues was essentially done using manual methods. But subsequently, rapid, easy to handle kits were introduced in the market for isolation of high quality genomic DNA. However, in tree tissues which are very high in phenolic content, isolation of high quality genomic DNA still remains a challenge. The commercial kits which are presently available in the market are not customized to handle the challenging tree tissues. Hence, the Institute of Forest Genetics and Tree Breeding developed a cost-effective kit for isolation of nucleic acids from these challenging tissues, which included the wood, the silica dried samples, the leaf samples, the root samples, and so on. The protocol was patented in 2009 by the Institute along with the Department of Biotechnology. ArborEasy, a trademark, was registered by the Institute to market the kit. The efficacy of the DNA isolated from several plant and tree tissues was validated by several national laboratories, and the quality of the DNA was checked by commercial R&D laboratories. The kit was launched on 24th January 2018. The advantages of ArborEasy DNA isolation kit are several. First is that it can handle the challenging tree tissues which the other commercial kits do not give very good results. It is a rapid procedure which takes about 1.5 hours to isolate high quality genomic DNA. It's also free of biohazardous chemicals. The kit is also cost effective and it is about 1/3 the cost of the existing kits in the market. The components of the ArborEasy DNA isolation kit include extraction buffer A, extraction buffer B, binding buffer to bind the DNA to the column, wash buffer A and wash buffer B to wash the column, and elution buffer for eluting the DNA from the spin column. The kit also provides spin columns for isolation of high quality genomic DNA. Isolation of genomic DNA using this kit is very similar to the available commercial kits. The protocol involves grinding of the tissues in liquid nitrogen and then transferring the ground sample into a preheated extraction buffer which is maintained at 65 degrees centigrade. Subsequently, the mixture is vortexed rapidly and centrifugation is done to remove the debris. The supernatant is then collected in a fresh microcentrifuge tube and chilled isopropanol is added to precipitate the genomic DNA. Subsequently, binding buffer is added and the mixture is loaded to the spin column and centrifuged. The column is then washed twice with the wash buffers to remove the contaminants. Now the DNA is eluted from the column using the elution buffer. The genomic DNA, so isolated, is now ready to be used for all downstream processes. The genomic DNA isolated using the ArborEasy DNA isolation kit can be used for several downstream processing. It can be used for routine PCR analysis like the RAPD, ISSR or AFLP analysis. It can be used for high throughput marker analysis like SNP genotyping, SSR genotyping, indel genotyping, and also the DNA is pure enough to conduct the whole genome sequencing and target capture. The kit has been used to isolate genomic DNA from an array of tree species like from eucalyptus, casuarina, sandalwood, red sanders, teak, melina, melia, ailanthus, to name a few. The customized kit was used to create the Guinness World Record in the India International Science Festival in 2018, wherein about 550 students isolated DNA from banana simultaneously. The students belong to the age group of 12 to 70. This is one of the highlight of this particular kit. Additionally, this kit has been catalogued in the Kerafast Inc. e-commerce company based in Boston, USA. Presently, the institute is manufacturing and selling the kit to different R&D laboratories.